So good morning, students. In the last class, we had discussed about the single cell protein, what are the microbes that is involved in the production of SCP, and what do you mean by exact meaning of single cell protein, in which way it is giving benefits for the human beings. Okay, for the growth of the large number of plants with the high quantity or high content of protein. Okay, so this is about what SCP. So in the today's class we will discuss about the next topic, that is the tissue culture. What it is? That it is the tissue culture. Tissue culture. Okay, what do you mean by tissue culture? Here they had given the term the tissue and culture. Okay, in this method or in this particular process, what they are going to do is they are going to artificially grow the plants. Okay, artificially they are going to grow the plants on a medium, on an artificial prepared medium which contains the certain sources such as carbon, nitrogen, growth regulators, and certain other essential nutrients. Okay, that is known as the tissue culture. So first of all, tissue culture, this is a branch of biotechnology. What it is? It is a branch of biotechnology. Okay, technology, which is one of the modern branch where they are going to develop certain organisms artificially. Okay, especially that is the plant. Okay, it comes under the plant biotechnology where cells, tissues or organs in this culture method, in this tissue culture method, whatever the cells, organs, tissues will be there, where cells, tissues, organs are culture here. That is, they are going to of a plant body. Organs of a plant body are isolated. Organs of a plant body are isolated and then grown on artificial medium. Okay, on which it is growing? It is going to grow on artificial medium under aseptic condition. Under aseptic condition with controlled conditions like controlled factors like conditions of temperature and light. Okay, this is known as the tissue culture. First of all, what is tissue culture means? It is a branch of biotechnology, especially the plant biotechnology. It is a branch of biotechnology where the cells, any cells of the plant, the tissues, any tissue you can take of the plant, or the organs such as stem, root, flower, fruit, whatever the organs of the plant body is there, of the plant body are isolated. Isolated means that is going to removed from the plant body, then grown on the artificial medium. Okay, medium means, see, for the surviving of the plants, it requires the soil, water, sunlight, Okay, and temperature. These are some essential factors which are required for the natural growth of the plant. But here they are providing that all things, temperature, light, water, medium such as soil and all, they are providing artificially. Okay, as well as whatever they are getting the nutrients from the soil and water, minerals, salts and everything, they are going to give it artificially. In what? By in the artificial medium under aseptic condition they are telling. What I mean to this aseptic means that is the sterile condition. It should be free from the microorganisms. What it should be? It should be so that some other harmful microorganism or some other microorganism or microbes should not destroy that particular plant growth. Okay, that's why they had given under aseptic or sterile condition. Sterile means that is the absence of microbes or any other microbes which is not required in the growth of plant. Okay, under aseptic condition and controlled conditions. Whatever the temperature, light will be there, that is maintained. How much required, how much temperature is required, how much light intensity is required, that all things is under the control of what, of whatever the scientists who are going to perform the tissue culture. So here within a short period of time they are going to grow many number of plants in this by implementing this tissue culture method. Okay, not naturally, but it is going to done artificially, which is going to grow on an artificial medium. Okay, so next we will see here in this topic they had given what they are going to do means they are going to take the plant tissue or either the cell, either the tissue or the organ. They are going to cut it, then they are going to take for the what, tissue culture. So let's see what is that particular cell, tissue, what they are known as. Okay. So see here. The plant tissue or the tissue, here one term they had given that as the explant. What they had given? They had given as the explant. What I mean by this explant? Let's see here. Okay, what do you mean by explant in the tissue culture method means the plant tissue or organ, the plant tissue or organ excised and used up for in vitro culture. In vitro culture. See here, what is the first step in the tissue culture means that is they have to take an explant. What do you mean by this explant means see, for example, this is the plant. Okay, they will take, this is the plant, they will take any cell, either a cell, either it may be a meristematic cell, any cell from the plant body, they are going to take cell or tissue or organ. Okay, organ means the either they will take full leaves, leaf only, root, stem, shoot, anything from the plant body, that we are calling it as the organ. Okay, either they are taking the cell, tissue or organ. See, this is the cell, this is the tissue and this is the organ. Okay, like this they are going to cutting. Okay, they are going to cut it from the plant. For example, they are taking an organ. If they are taking the organ, for example, that organ, you will consider here it as the leaf. Okay, so this is the organ which is cutted, excised, excised from the plant body, and for what reason they are going to use? They are going to use for the in vitro culture. Okay, in vitro culture means that is growing, that is going to grow artificially. Okay, that is known as the explant. So this question may ask for 1 mark. What is explant? Or what is explant? This is very important question. The plant tissue or organ excised and used up for the in vitro culture. So this is known as the explant. Next thing here, whatever the explant, it may be the root tip, as I told you, it may be root tip, shoot tip, flower, leaf. Okay, any cell, you take any cell. So it may be root tip, shoot tip, stem, or embryo. In the flower, they will take the embryo, anything, any cell from the plant body, they are going to take that part of the plant which is cutted from the plant and taken for the in vitro growth, or which is taken outside the body for the tissue culture. That is, that particular part is known as, or that particular plant tissue or organ is known as the explant. Okay, next here, whatever the plant tissue culture method is there, it was first started by Gottlieb Heberlein. Who started this first, the plant tissue culture? Let's see here. See, listen here. That plant tissue culture method or plant tissue culture, it was first started by, or first discovered by a person whose name is Gottlieb Heberlein. Okay, the person who has first discovered this plant tissue culture, the name of that person is Gottlieb Heberlein. Gottlieb. Okay, Gottlieb Heberlein. In the year, in which year they had discovered? That is in the year 1902. Okay, so the most basic concept of the plant tissue culture. You see here, the most basic concept of plant tissue culture is, that is totipotency of cells. Okay, what it is? That is the totipotency of cells. Now, what do you mean by this totipotency? You will see here, totipotency of cells. This is a very important point. Please listen. Totipotency of cell, which was first, the term totipotency, it was first given by T.H. Morgan. Okay, who had given the term first given by a person whose name is T.H. Morgan. Okay, What do you mean by the totipotency, which is the basic concept of the plant tissue culture? See here, totipotency. This is also very important question for 1 marks. Totipotency means the capacity to generate a whole plant from any cell or explant. The ability or capacity to produce a plant from any cell, tissue or explant also we can say. The capacity of a plant, the capacity to generate a whole plant capacity to generate a whole plant from any cell, tissue or organ. So indirectly we can also say it as the explant. Okay, see here, whatever the plant they are going to take, if they are taking any cell, for example, they are taking a cell. This cell has the ability to grow a whole plant. From this single cell, they are going to grow a particular whole plant. So this cell, whatever this cell is there, that particular cell is having the ability to produce, to form a whole plant from their particular cell or explant also we can see. That property, that characteristic feature of that cell, we can call it as the totipotency. Totipotency is also asked for 1 marks. This is also 1 mark question. Please note it down. What do you mean by totipotency? Means the capacity to a whole plant from any cell, tissue or organ, or else you can directly write it as the explant. Okay, that is known as the totipotency. So see here, what I told, what is the method of tissue culture? This is about some terms. So first they are going to cut the any part of the plant, or they are going to take they are isolating the explant. The next thing, after isolating, they will wash them. See here. First thing, they will take any, this is the plant. This is the plant. Okay, a selected plant for the growth of the same plant. So here, they are going to take any cell, tissue or organ, to which we are calling it as the explant. Okay, they are cutted from the plant and the tissue. For example, that explant is the leaf. You are taking a leaf. So what you will do means, after taking that explant, you will undergo into a process that is known as the surface sterilization. What it is? That is the surface sterilization. What do you mean by this surface sterilization? Means they are going to, that explant are surface sterilized by certain disinfectant. They are going to clean with water. The leaves, that is going to clean with the water. Okay, they will keep these leaves so that all the dirt should come out. Okay, then they are going to use some disinfectant, such as ethanol or alcohol they are going to use. So it should be free from the microbes, which may damage the cells in the further process. The next thing, What they will do? They will prepare the nutrient media. Preparation of nutrient medium. So, whatever the nutrient medium they are going to prepare, what does it contains? Let's see. That medium, whatever the medium they are going to use, it should have a sugar content, or else this medium should have this nutrient medium. Okay, see, for example, they are preparing in this beaker. First, they will use. Okay, first they will add here, that is the sucrose. What they are going to use as a sugar source means that is the sucrose. First thing they are going to add the sucrose, then which is this sucrose is a carbon source. What it is? Sucrose will act as a carbon source. After that, they are going to add some of the inorganic salts, vitamins, and a very important point. After vitamins, they are also going to add the amino acids. A very important point. They are going to add some of the plant growth regulators. What they are going to add? That is the plant growth regulators. And what are the plant growth regulators? We can also say it as the plant hormones. Okay, growth hormones. What it is? That is the growth hormones. Okay, which hormones they are going to add in the nutrient medium means that is the auxin and cytokinin. These two hormones they are going to use, or they are going to add. First thing, they are going to add the sucrose, which will act as a carbon source. They will add some inorganic salts, vitamins, amino acids, and plant growth regulators for the preparation of what? For the preparation of a nutrient media or a nutrient medium. On these, and as well as they will also add. See how the soil is the base, a particular medium for the growth for the plant. Here they are not going to use the soil, but they are going to use one of the gel substance, that is the agar agar. They are going to add the powder that is known as the agar agar powder, which forms a medium for what? For the growth of the. Okay, so this is about the preparation of what? Nutrient media. Okay, see next we will see. See after that, what they are going to do means they will inoculate into the particular medium. After preparation of media, inoculation into the medium. Okay, either they will take a petri dish. In this petri dish, they are going to add the medium, okay, as well as with the agar agar powder, which will form a base, a medium for the growth of what? For the growth of the plant. So what they are going to do means, whatever the explants they had taken, this explant they are going to inoculate there in the medium. Inoculate means they are going to put there in the medium like this. Okay, this is known as the inoculation into the medium, inoculation of explant into the medium. Okay, so next, so till there you had come to know. Next we will see. Next thing, what they are going to do means next, what is the next term which is used over here means that is micro propagation. I will explain that one. That is also very important. See here, by the application of tissue culture, okay, tissue culture is however helpful for the plant growth. See, by the application of tissue culture, by the application of what? Of culture, it is possible to achieve a propagation of a large number of plantlets. Okay, large number of plantlets. Wait a minute. Plantlets in a very short duration. Very short duration. Okay. So in a very short period of time, they will get many number of plants or a plantlets, what we call. So that particular propagation or the production of many number of plantlets, this method of production, how many means it can go up to the thousand number or so this method of production of production or propagation also you say production of thousands of plantlets, okay, in a short period of time, okay, through the tissue culture, and this method of production of thousand number of plants, that it is known as the micro propagation. What it is known as? It is known as the micro propagation. Okay, what it is? That it is the micro propagation. Here you see, in tissue culture, by the application of tissue culture, it is possible to achieve a many number of plants. It is possible to produce a many number of plants. Propagation of large number of plants. Large number means the number will go up to the thousands. Okay, that in a very short period of time. In a very short period of time, at a time you can produce a many number of plants, and that production of thousand number of plantlets through the tissue culture, it is known as the micro propagation. What it is known as? It is known as the micro propagation. Okay, so here, what are the advantages of micro propagation? We will discuss here. This is also very important point. What is micro propagation for 2 marks or 1 mark? What are the advantages of micro propagation? We will see here. Wait for a second. So, what are the advantages of micropropagation? We will see here. Advantages of micro propagation. Okay, so in its advantages, what are the advantages of micropropagation? Means the first thing, it is easy, safe, and economical. Easy, safe, and economically economical method of propagation, or for the production of many number of plants. Then it helps in rapid multiplication, because there is a very soon, in a very short period of time, you can see many number of plants are growing. So helps in rapid multiplication of plants within a short period of time. Okay, the next here, plants can obtain throughout the year. There is no particular season, because there is no particular natural temperature, natural light. Okay, they are going to provide artificially in the labs. So it is a plants can be obtained throughout the year. Plants can be obtained throughout the year. This is one advantage. Next, sterile plants can also be multiplied, and as well as ornamental plants also be multiplied, and there will be growth of flowers in a very large number. And rare and endangered plant species also can be grown by using this tissue culture method. The next, this is about the micropropagation. Next here, next term comes, that is the soma clone. What do you mean by this soma clone? This is also very important. Let's see. Soma clone. This is also very important. Listen here. Each of this plant is genetically identical to the original plant. Each of these plant is genetically identical to original plant, from which it is derived. Okay, see, there I had shown you the inoculation. After inoculation, what happens means there is an formation of callus. Callus means a mass of cell is going to form in the Petri plate. See, for example, this is a Petri plate, means a callus is going to form. They will remove that callus and then they are going to culture into the beaker which contains the media, Okay, they will subculturing they are going to use and you can see certain mass of cells are going to grow and that is going to and here this mass of cell which is known as the callus this will give rise to a small shoot and a small root system that is in the test tube or in the beaker after which a new plant is going to form and whatever the plant which is going to form new okay or a new plant which is going to form over here this plant will be identical that means similarly to the parent plant from where it has taken or X plant from which plant they had taken they are this new plant will be similar to that plant so that is known as the soma clone means copies they will be exact copies of their parent plant so each of these plant which is genetically identical not only the phenotypically or you can see not only the morphological characters are same in the plant their genes they are genetically identical to the original plant which from which it is derived okay so that is known as the soma clone the next one more term is there that is known as the somatic hybrids and somatic hybridization we will see that one which is the last topic. See here, in the somatic hybrid, for the production of somatic hybrid, what they will do means the process of first we will see what do you mean by this somatic hybrids. So scientists have isolated protoplast. What do you mean by protoplast means they will take the cytoplasm with the plasma membrane. Okay, so here first we will see what do you mean by somatic hybrid. Somatic hybrid. Okay, this is also very important question for 1 mark or 2 mark. What do you mean by the somatic hybrid means that it is a hybrid formed by the fusion of somatic cells of two varieties of species. It is a hybrid which is formed by the fusion of somatic cells of two different varieties or species of two different varieties or species. That is known as the somatic hybrids. How they are going to see? First, what they will do is first thing they are going to isolate the protoplast. They are taking the protoplast of two different cells. Okay, they are going to take isolate the protoplast. For example, this is the protoplast of one variety. Okay, and here there will be presence of nucleus and other cell organelle with cell membrane or cell wall. Okay, and as well as there will be presence of cytoplasm. This is one somatic cell. This is somatic cell 1, and here this is the another somatic cell. Okay, somatic cell 2. What they will do is first thing they will isolate the two different somatic cells from the two different varieties. Okay, for example, we can take example as the one is the tomato and another is the potato. Okay, they are the two different varieties or two different species. They are going to take the two different cells. The next thing they will see each whatever the varieties they are going to take, each of them will have a desirable characters. So these characters can be fused. they are going to fuse it. How means first before fusion, what they will do means they will digest. The second step, they will digest the cell wall. Cell wall. How they are going to digest the cell wall means by using an enzyme. Which enzyme is that is cellulase and pectinase. Now the cells are free from the cell wall. Okay, how? That is by using the cellulase and pectinase, they are going to remove the cell wall or digest. That cell wall is going to digest it. Okay. Now next here you can see. Here I will show you. This is the one cell without cell wall, and this is the another cell without cell wall. They are going to fuse by using one of the substance that is known as the polyethanol glycol. By fused with the by PEG, that is polyethanol glycol. What it is? PEG, that is the polyethanol glycol. Then these cells will get fused like this. This is the cell fusion. The two somatic cells are going to get fused. Then next there will be a nuclear fusion is going to take place. This is the nuclear fusion. After that, a hybrid is going to form with the two different varieties, with two different desirable characters. You can see a hybrid, and this hybrid is known as the somatic hybrid. What it is known as? It is known as the somatic hybrid, and this process for the production of this somatic hybrid, it is known as the somatic hybridization. This question may ask for 2 marks. That is, what do you mean by this somatic hybridization? Okay, so this is about the soma clones, somatic hybridization, somatic hybrids, then micropropagation. One example I told you, that is by fusing tomato and potato, they will get a new variety, new hybrid. The name of that hybrid is the tomato. What it is? That it is the tomato which is not commercially used because they don't have such desirable characteristic features. Okay, so this completes your chapter about the strategies for enhancement in the food production. Okay, so this completes your chapter. Then next, in the next class, I will go with a new chapter. First, I will discuss some of the questions based on these chapter. Okay, and the next chapter is microbes in human welfare. Thank you.